The broadcast is now starting. All attendees are in listen-only mode.
Hello everybody, welcome to September's Global Health Drug Design meeting. I hope you all had a good summer, and I'm very pleased to welcome back Antoine Daina and Vincent Zoete from the molecular modelling group at the Swiss Institute of Bioinformatics. Those of you who attended June's meeting will have heard their first presentation where they were talking about their new ADMET server, and they introduced some of the other packages that that Swiss Institute for Bioinformatics also hosts on the website. And today they're going to give you some more details about these, particularly those to do with docking, virtual screening, target prediction when you've got the results of a phenotypic assay, and some more of their drug design tools. I hope you enjoyed the presentation, and I'm going to hand over to Vincent and Antoine now. Okay.
Thank you very much, Caroline. Thank you very much for attending this new webinar. So I am Vincent Zoete, and I will present briefly different theoretical introductions regarding our different tools. And then Antoine will present the most practice exercises that will follow. Yeah, the SwissDrugDesign project is based on the typical drug design pipeline that you see here, which, no surprise, go from target prediction to clinical development through hit finding, hit to lead, lead optimization. There is no surprise here. And actually, the purpose of the SwissDrugDesign project is really to help designing and answering questions along the drug design pipeline, notably around the hit finding, hit to lead, and lead optimization processes. So we have developed different tools. So actually, SwissDrugDesign is not a tool itself, it's a name of the project, and this project is composed of many different complementary tools that try to address those different questions using structure-based approaches. So everything that you can do when you know the three-dimensional structure of the targeted protein, and ligand-based approaches, which is everything that you can do maybe when you don't know the structure, but you have access to a series of molecules that are already active against the target. So we will make a very fast overview of the different approaches, and starting with the structure-based approaches. And at the very beginning of the project, actually, we developed a docking software. So you know that the purpose is to find the most probable position of a small molecule at the surface of the protein. And typically, a docking software is composed of two components. The first one is a sampling engine that tries to find possible positions of the small molecule at the protein surface, of many different possibilities. And you have a scoring function which tries to estimate which of these different possibilities is actually the most likely. So for the scoring function, we try to use something which is physics-based, universal, so that we can not only apply it to protein engineering, but we also demonstrated that we could apply it to drug-like molecules, molecular fragments, lipids, peptides, without any modification, which is very useful for us. So based on this, which is relatively time-consuming, we actually developed different docking engines. Typical one based on evolutionary algorithm, or more dedicated one like the reconstruction of the small molecule by dihedral angle sampling that you will see in the next slide. So we had two main focuses. The first one was to get a very good success rate, so to be able to reproduce binding modes when they are not experimentally, and also to diminish the computational time required to do the docking, so that we could put everything behind the web server, as you will see. So the idea of the last docking software that we developed, DSS, is actually to first identify the different cavities and grooves at the protein surface, and then the sampling engines actually position the center of the small molecule at different well-chosen position of these different pockets, and we reconstruct the rest of the molecule. So, for instance, in this image that you see here, you can see the process when you have already reconstructed one part of the molecule, but you need to reconstruct the second half of it. So the idea is that, for instance, we will first position the atoms that are in red here, so this amide function, and we make a dihedral angle sampling of the different position possible for this oxygen atom and the nitrogen atom by rotating around the dihedral angle. Once we have found good positions for these two atoms, we will connect the next one of the molecule. So for instance, there's two carbons. Again, we make all possible rotations to find the best positions, and we go ahead and ahead until you reconstruct in entirely the small molecule in the binding site. So that's the basics of the algorithm. Of course, it's more complicated than that, but I mean, the full demonstration is out of the scope here. So the idea was to increase the speed of the process so that we can make a simplified website and provide access to that type of tools also to non-specialists in the field. You will see with Antoine that you don't even need to provide the computational power and install any software. Everything is actually done online, and we compute on our own machines automatically. So I will not detail that. Antoine will do it later. So here I wanted just to mention two additional tools. So those ones are really dedicated to specialists in the field, people making molecular modeling. The idea was to provide everything you need in terms of topologies, parameters, different data like that, to be able to perform computer-aided drug design using molecular mechanics packages like CHARM and GRUMAX for organic molecules, but also non-natural amino acids that are commercially available. So this is really for specialists in the field. There is no web tool to use, so actually I will just keep that for the moment. And now I will concentrate on the ligand-based approach tools that we have developed. So the first one actually was Swiss Bioisostere. So here the name might be a bit misleading. So we are not just concentrating on bioisostere, but on any kind of molecular replacement. So the idea is, as you know, is that in lead optimization, we spend a lot of time modifying molecular fragments at the molecule to address different issues regarding maybe the affinity, ADME problem, or an intellectual property issue. And those molecular replacement actually comes from the experience of the medicinal chemists in the approach of the modelers, and everyone is very good at it generally. But we wanted to bring something more comprehensive to the field, and this is the reason why we developed this tool, which is actually a database of possible molecular replacements. So for this, we started from what you see here on the left of this slide, which is a library of bioactive molecules. We extracted all pairs of molecules that are similar, that are active on the same target, tested in the same assay by the same team, published in the same paper. And when we find such a pair of molecules, then we can extract, using different algorithms, the difference between those molecules that you can see in the middle here, for instance. And those differences are actually the molecular replacements between the two molecules, so that we can store this information in a database along with the complete molecule, the target against which they were tested, the assays, the results, so on and so forth. And we provide this through a simplified web interface, as you will see with Antoine. So we did that for several hundreds of thousands of molecules that we find to be active and well determined in the Campbell library. We could isolate this way about 20 million molecular replacements in the medicinal chemistry literature, including 4.5 million that were unique. So actually each of these unique replacements has been maybe just tested once, and we can find it out, but some actually have been tested tens, hundreds of times, maybe thousands of times, and we have all this information that you can retrieve automatically for lead optimization. So as you will see with Antoine, if you want now to replace a molecular fragment in your molecule, you can just draw it in to the sketcher of the website, tell us where the rest of the molecule should be connected, and you get a list of possibilities that have all been tested experimentally. For instance, for this particular replacement, we can provide thousands of replacements. For each one, we know in which molecule it was tested, against which target, what was the effect on the activity, physicochemical properties of a small molecule. You have plenty of information in statistics that you can access. You will see that with Antoine a bit later. So something else that we can do in ligand-based approach is to capitalize on molecular similarity. So the idea here is that if two small molecules are very similar to each other, they are more likely to be active on the same target. And so to use this assumption, we need to define what a molecular similarity is. And in this field, we have two main definitions. The first one is based on the chemical structure. So you see here two examples of EGFR inhibitors, and we immediately see that they are similar because they are based on the same scaffold. So the idea of the computational tools is actually to be able to do that for millions of pairs of molecules, and also to quantify the similarity so that we can actually rank the molecule based on their similarity afterward. So To work on the two-dimensional, so on the chemical structure similarity, we can use different techniques like fingerprints, so I will not detail this, but we can really treat about 1 million pairs of molecules per second using this approach. So it's something which is very powerful to detect chemical similarities. The second definition that we can use is the similarity based on the shape. So here you can consider, as you can see in the bottom of the screen, two molecules are similar if they have the same shape, and if they project charges and lipophilic group at the same position, so that they will occupy the space of the binding site the same way, maybe with a totally different chemical structure. So this is slower than the first approach, but it is also very good to potentially make scaffold hopping, so finding molecules that are both active on the same protein, but potentially with totally different chemical structures. So if you can quantify this way molecular similarity, you can do different activities. The first one is to make virtual screening. So the idea is very simple, and you already know it. I mean, if you have in your hand a molecule which is active against a target of interest, you can screen a library of molecules, maybe with a known activity, so just commercially available molecules, for instance, for those that are similar to use. And if you can find molecules with high similarities, and you can test them in priority because they could be active also on the same target. So for instance, if you screen the library of Campbell of 2 million molecules, also for those that are similar shape, to nilotinib, you can find different molecules that you can see on some of the examples on the right that are also active on the same target, which is in this case the ABL kinase. So this is something that can be very powerful. So the problem with screening is that also it's conceptually simple and very fast. I mean, the calculation itself just take a couple of seconds, 2 minutes. The technical burden is to prepare the library for the screening, because you need to take the library, remove solvents, remove ions, check the tautomers, check the protonation state, make three-dimensional conformation for the small molecule. So you have many different technical aspects for the preparation of the run. And this actually is a bit annoying, but the idea of Swiss Similarity is to provide access to these screening tools by preparing once for all those different libraries to be used with different types of approaches to determine the similarity. So that now, with our website, you can screen a library of drugs or developmental drugs, bioactive compounds, commercially available compounds, and we also made a library of virtual compounds to increase the chemical space accessible to screening. So the idea is that now you can really screen the different libraries for your molecule using different approaches just upon some clickings. And if you find molecules that are similar to yours, you can start additional calculations using SwissDrugDesign tools, or we can provide access to the original database where you can find and maybe purchase a molecule. For this library of virtual molecule, we wanted it to be useful, so not just a collection of impossible molecules, but something which is really accessible. So we started from the sigma-rich library of compounds, and we applied virtually a series of click chemistry reactions to them in a combinatorial way. So we created 1.2 billion different chemical reactions, and we screened the different reactants to only keep the drug-like ones according to different chemoinformatics filters. And this gave this library of 200 million molecules. So this means that each single molecule in this library is potentially synthetic accessible by click chemistry reaction from commercially available fragments, and we can provide the pages where in sigma-rich you can check the availability of the molecule. So of course we cannot be absolutely sure that each single reaction will work very well with a different fragment, but at least we increase dramatically the chances that the molecule can be synthesized. So again, we have made a simplified website that you will use with Antoine, where you can just provide the molecule of reference and make the screening very rapidly. Another usage of molecular similarities that we can do is to do exactly the reverse of screening, so to predict the target. So the idea here is that, as you know, the assumption that the activity of a small molecule is explained by the binding to the primary target is not exactly true, because some of the FDA-approved drugs have unknown targets. So we just know that they are bioactive, but not why. And for the rest, we know that there are several possible targets to which they can bind, and those bindings can be responsible for some of the side effects that can be connected to toxicity, but also to drug repurposing, which is a very nice aspect today. So actually, it is very important to be able to predict the targets. And again, we relied on the assumption that two molecules that are very similar are more likely to be active on the same target. So what you see here, the blue curve, correspond to the probability that two bioactive compounds are active to the same target as a function of the shape similarity. You can see that above a given threshold of similarity, about 90% in this case, we are able to make nice predictions regarding the possible targets of a given molecule. So this curve was obtained by making calculations on a library of 350,000 molecules for which we know exactly the target. So the idea of Swiss target prediction is actually that if you provide the website with a small molecule, we will automatically screen this library of molecules for which we know the target, find out those that are the most similar to your compound, and from this, we are able to calculate some probabilities with which your molecule could actually bind to those targets. So what is true here for shape similarity in this curve is also true for the chemical structure similarity. And in the lab, we actually decided to conjugate both scoring function in the same approach, so a logistic regression that allows us to make predictions not only when the molecule belongs to a series which is already well known, and so we can capitalize on structure similarity, but we can still predict something interesting even if you are working in a totally new family in terms of chemical structure. As you can see here, for molecules of a drug-like size that have very new in terms of chemical structure, the combination of the two scoring functions allows us to make relevant predictions in difficult cases also. Some aspect which is very important with Swiss target prediction is actually the possibility to make predictions in different organisms and not just in humans. So the idea here is that if you are working in a molecule for instance, a human target, as you can see in this example, you may be in a situation where no molecule similar to this one in terms of chemical structure or shape has ever been tested against a human target. But you may find examples where a similar molecule has been tested on another organism, like rat in this case. And thanks to this, we can actually make the prediction on the human target. So we have made statistics to see whether this orthology relationship is actually working. And the conclusion that it actually works. So as you can see here with the different curves, if you compare the gray bar to the green bar where we are using this orthology relationship, you see that we can dramatically increase the success rate with which we can determine the structures here, notably for rat, mouse, and cow. So it's not working with human because we have so many information and active molecule for humans that what we have tested in rat, mouse, and cow actually is not helping, but the reverse is actually true. So actually, if you are working on an organism for which not a lot of molecules have been tested, you can capitalize on the different targets you can find in other organisms in our approach. So this is not only working with human, rat, mouse, and cow, but with whatever other organisms that you can see. Also, we dig a bit further and we see that if this already works, if you have a molecule which is similar tested in another organism, if it is the very same molecule that has been tested in another organism, then the prediction is even better, and it's even increasing the prediction in the case of human targets in this case. So actually, orthology is something that is very useful to make predictions throughout different organisms. Again, we have made a website that you will use with Antoine. You can draw the molecule of interest and we can predict different targets. So you will see that in details in a couple of seconds from now. And also just a reminder about SwissDrugDesign. So we made a webinar before summer that is still available. So if you want any details about SwissDrugDesign, actually, you can still have a look at this webinar and you will find many information. So today we will not address this one, but it's still available, of course. So one last aspect that I would like to underline before Antoine will start the exercises is actually the fact that we plan to make those different tools integrated and interoperable, which means that the output of a tool will be able to be used as an input for the next tool, so that you can really create a computer-aided drug design pipeline on the fly without any technical burden, without never downloading a file, uploading a file, or making file translation between the tools. Everything will be automatic. It's already in place for the newest tools like Swiss Similarity. So for instance, here you see an extract of the Swiss Similarity output page. You have icons next to every single molecule in those output pages. If you click on the pills and you transfer automatically the small molecule to Swiss ADME and you get the results after a few seconds, which means that to get the ADME results of a given molecule in another tool, you just need to click on one icon and wait a few seconds, so it's very convenient. So with this, I would just like to conclude and remind you that actually all the tools that you see here are totally free to be used for academic research and not-for-profit research. It's also login free so that you have no technical burden again. We also try to make our tools easy to use so that we don't need to be specialists in the field to use those tools. They are providing access to different approaches that are unique in the field, like this database of molecular replacement, different scoring functions for screening, the ADME tools that you have seen last time. It's also available in the long term because we have the support of the Swiss Institute of Bioinformatics to maintain those tools in the long term and provide them for free for academic and non-profit research. So actually, the tools that you see here will still be available in several years from now and will not be closed. To finish, I would like to thank the many members of the molecular modeling group that work in this project, notably Antoine, that will now take the lead to show you the exercises.
Great. Thank you, Vincent. So for the rest of the webinar, the idea is to use a fictitious exercise to introduce as a support a few concepts in the use of our websites. So as a preamble, I have to say that we are not working in neglected disease, and we're actually not working in infectious disease. So we are not in the field. So maybe some of the molecules I'll show have some issues or are not really suitable for this specific purpose. But it's really trying to have a support to use our site. So let's say that the exercise is we perform a phenotypic screen, and out of this phenotypic screen for malaria, a molecule pops up as a hit, which is this one. Let's say, so this is the structure of the hit. And we can ask 3 questions. One, of course, is which other molecules can I buy, can I purchase, can I obtain and test to increase the number of potential hits with different chemistry or very related chemistry? What could be a possible target protein for this hit? And of course, how can I generate ideas to make design, so a small chemical modification, either for hit expansion to lead or for lead optimization, right? So yeah, really, all our tools are through the web, right? So all these are website. They are, as Vincent said, absolutely logging free and free for nonprofit. One thing is that we tested extensively these sites for Chrome, so Google Chrome browser and Firefox. So of course it can work with other browser, but the best experience surely is with these 2 browsers. So simply, if you open a browser and you type the URL of the site, you have it. So we will start with the ligand-based screening tool to try to see how we can get new molecules with this hit. So SwissSimilarity.ch is this website. All our websites have the same look and feel. There is very often a sketcher to draw a molecule and a text box to enter the smiles, right? So if we want to input a molecule, you can draw it, of course, with a very standard sketcher, the one of Chemaxon. Respect to time, it's very easier to copy and paste a molecule. Of course, you can also import a file you have on your machine, or you can be connected to DrugBank or other structural chemical databases. Only typing names or UPAC definition, you can import a molecule. So here, this is the hit we have from the screening, and the first question is, let's try to see which other compound can be purchased. And for this, of course, the zinc database is the best choice. It's a compilation of vendor catalogs that are curated for drug design. So in Swiss Similarity, you have to find the database you want to screen. Here it will be the zinc lead-like molecules, so more than 4 million lead-like molecules we prepared already for screening, and you have to choose a method. So there is FP2 fingerprints for chemical similarity, electroshape for 3D, and other more time-consuming 3D methods like pure 3D alignments. And also, Vincent showed you, there is the combined score, which is a logistic regression of FP2 and electroshape. So let's try to screen the zinc lead-like compounds with FP2, which is the one that is faster, 10 seconds of pure calculation, right? So with clicking this radio, it's really the method and the database, and then this button is red, so you can click on it. So normally in a few seconds you will have the output of this screening, if everything goes well. Right, it has started. So sometimes when you have the server that is not fully available, you have to queue a little bit, so wait a little bit. Okay, this is the output page, right? So this is the query molecules, and these are ranked by Tanimoto score on fingerprints, so on chemical similarity. We have these molecules here. So honestly, here with chemical similarity, what you can get is not tremendously interesting. So you have this part here that look like the right-hand part of the molecules, which is maybe not something you will try to purchase to test in your screen. You are looking maybe for something like is more sensible compared to this one. So you will run another screen on the same zinc lead-like collection of compounds. So for this, you will click on this little twins icon. So it opens a new tab. Normally it opens a new tab, yeah. And everything is already filled with the molecule you want to screen. So again, we will screen the lead-like molecules of zinc, but now we will use electroshape, right? The problem of this is that you have to generate the 3D and align the 3D, so it's 7 minutes. So it's a bit long for the purpose of this webinar. So I already run the screening actually, and it shows you that you can simply copy paste the result, the URL of the result page, and you get it. So it stays on the server at least 1 week. So you can export to make reports, but you can also, for instance, email the URL so you can get back on the result for 1 week. So here it's the lead-like collection of zinc, but no more with 2D similarity, but with 3D similarity. And you can see that here, maybe the output is more interesting. Basically, you have 3 family of compounds that look like similar in 3D shapes and projection of charges and lipophilicity. You have this kind of compound that chemically are pretty different from the hit. So it can be very interesting to get this one for seeing if you can do SAR, you can change a bit the chemistry and have very different hits. You have this kind of molecules that are much more similar to the input molecules, but what is interesting is that in this family you can see you change the right hand side and the left hand side, really the extremity of the molecule. So potentially if you test this molecule, you have information on expansion of this part of the molecules. And a third family, that is this one, interestingly, you can rigidify the compound. So if this compound is lead-like but a bit too flexible, you have some opportunity to have less flexible compounds. In any screening, you can you have a link to the original database. So here you have a link on zinc. So if you click on the name here, you open the corresponding zinc entry. This is the molecules, and what is interesting here is the vendor. So you have the provider where you can purchase this compound. Same for the others. So you can see here, for that one, you can go to this company that is tetrahedron building blocks, and the same for the third one, and I think it's the same provider, yeah. So it can be also a criteria selecting the molecules also on the availability at the different vendors, right? Okay, another interesting thing we can do is what Vincent told you. So you can browse drug, you can search for similar compound, bioactive compounds, like for instance in the Campbell database. We have seen that you can look for commercially available compound in the zinc, but also virtual library. So we have generated this almost 300 millions of compound library, which is obtainable by click chemistry with building blocks you can find at Sigma Aldrich. So here, of course, 3 million compounds, even if it's with fingerprint, it's almost half an hour, right? So again, I have run it. Before, and I have saved the URL. So this is the result. So you can see here that you have similar compound in terms of chemistry obtainable. So if you are interested for whatever reason in that compound here with this kind of steroid moiety, you can click again on the link, and you don't have directly access to a database, but here you have access to the reaction we used to generate these virtual compounds for these two building blocks that are at Sigma Aldrich. So, and you have a definition, so it's a reductive amination here to get this compound. If you click on the link now, you have access to the catalog. So this is Sigma Aldrich website. This is the compound or the building block or the reactant you want to buy, and you can already have an idea of a price. So for half a gram is $280 here. So you can go to that kind of detail. If we go back to Swiss Similarity, the last maybe interesting database to look at is the ligand from the PDB, right? It's, you will screen the ligand that have been co-crystallized in protein target. So potentially it can bring a lot of information, right? So this is the screening. So you have 20,000 compound to screen, so it's almost at a click. So these are the results, right? For FP2 fingerprint similarity. So at 0.8 similarity, for instance, you have this one. This is the same compound with different stereochemistry here. This is the most similar to our input. So if you click now on the link, you access the entry in the PDB for that small molecules. So you have a lot of information, among which you have the PDB entries for co-crystalline complex, including these small molecules. So I already checked a bit, and I have found something that is potentially interesting for us here. It's this molecule has been co-crystallized in this alanyl aminopeptidase here from Plasmodium. So potentially interesting, right? In our context, this is something that we have to keep in mind, right? So if we go back to our exercise, we have kind of answer question 1, screening zinc. We have targeted some catalogs where we can find interesting compounds that are similar in 2D and in 3D. The next question is, what is the potential target, right? Which protein is most probably target in this hit we get only from a phenotypic screen? So for this, we have a tool that is Swiss target prediction, which is performing ligand-based reverse screening, as Vincent told you. So now is the same concept of molecular similarity in 2D and in 3D, but we screen against compound that we know the target. The target is in the literature and is well defined. The bad news, if I can say, is that we are not in the neglected disease or infectious disease area. So we mainly focus on protein targets for human, right? Which is not perfect for our purpose, but actually, as Vincent have shown you, it's possible to deal with homology. So as Vincent explained to you, homology and orthology helps you if a compound is similar in an ortholog or a homolog protein, it's also a way to define the target. So here I have run automatically in human the prediction entering the smiles. You can see that we have human, we have mouse, we have rat, we have a cow, and we have horses. So not that we are particularly interested in veterinary medicine, but it's for this question of orthology. So in less of a minute, you have the result page. So this is the query molecule. We look at similar molecules with known targets in Campbell. This is the first general result, giving you the target class of all predicted probable targets. So 2/3 of the targets are proteases, 20% are GPCR, and the rest is enzyme, right? And here on this table, you have the actual prediction ranking. So the most probable to the 20th or something less probable target. Probability is represented by this green bar, which is the logistic regression of the score of 2D similarity and 3D similarity with different molecules. You have also the common name. You have a link to UniProt. So if you are interested in this aminopeptidase M in human, and you click on this link, you have a link to UniProt, SwissProt entry, and all what you need concerning this potential target. You have also a link to Campbell, and you have a possibility to look at what has driven the prediction 2D molecules that have been measured active on this target. These are the molecules, and you can see Tanimoto score for fingerprint, and the same for 3D, right? And you have the score for electroshape superimposition. Right, so as I told you, for human target, we can use homology directly, like this one was predicted by homology. In the case of malaria, we have to do to look for homology protein ourselves, right? So let's say that we go for this aminopeptidase N, which has this UniProt code here, and we will simply blast for Plasmodium falciparum molecules, right? So it's very a simple blast alignment. So I go on the NIH blast, which is absolutely not our tool. We will screen only Plasmodium falciparum sequences this way. So it will take a few tenths of seconds here. It's running. So really very simple sequence alignment of protein sequence here. Right, and this is the output. As you can see, you can find decently homologous protein in Plasmodium falciparum. Actually, all the first ones is the same aminopeptidase from malaria, as you can see, and the difference is this is different PDB entries, so you have different complexes, but everything is the same, right? So in a few clicks, you have the PDB entry for this homologous protein, and what we can do is make a very simple docking, right? So we take our molecule and we look at if it's possible to dock our compound into this crystal here, and this will to gain confidence about the possibility of this one being a possible target for our hit from our screen, right? As Vincent said, we have our web-based docking engine, which is SwissDock. Information here, you click on the tab for submit docking, and you have the possibility to enter the name of the protein, like this aminopeptidase, and search for it. So we have access to different database. You can select the specific molecules and so on and so forth. Respect to time, I already prepared the molecules, the protein in a PDB to gain some times and not let the process automatically make the preparation for you. Same for the ligand. Let's say that you have screened the catalog of a vendor, you have the zinc ID. Here I have prepared the molecule, which is my molecule, with the hydrogen and everything, right? So it takes less than a minute to set up the thing. You have to put a name, my molecule in this. It's good to make to provide an email address, because as it is pretty long. Here, for instance, it's a big molecule, and we will make a blind docking. So trying to dock the molecule on the entire surface of the protein. So it's pretty long, so it's multiple hours calculation. So if you don't want to let your browser open, you can provide an email address that tells you when the docking is finished with the link, and you can simply click on it and get the result. you have extra parameters. You can increase the speed, which is not in our context, not truly recommended. You can increase the flexibility of the protein, which is only experimental at the moment, so not really a mature way of doing flexibility. But what is can be interesting is to perform focus docking. So if you know where is your preferred binding site of your molecule, you can define a zone, a smaller zone that the entire surface of the protein, providing coordinates and the size of a box. And then the docking is not only much more fast, but also much more precise, because you make multiple assessment in the same cavity. So it's not the case for us. We will start a multiple hour docking, clicking on this link. So of course, as I told you, I've already run the docking, and I will simply copy paste the link that I have received on my email. So after a few hours, you receive an email with a link, and you got this access to this page, which provides you with a very simple 3D session here with the protein. So you can see here the secondary structure, the tertiary structure of our aminopeptidase in Plasmodium. And here you have the list of docking poses. So here maybe it's very difficult to see. Here there is in the middle here in the first cluster a result really at the center of the protein. Here you have another, but here at the surface of the protein, so on and so forth. Sometimes it's already sufficient to have this kind of analysis, but of course, when you need more detailed analysis of your docking results, you need to open it in visualization software package. So two options to do so. One is if you have a UCSF Chimera visualizer installed on your machine, by clicking this link here, you open directly the docking. If you want to use, or you must use another visualization package, you can download all the files in very standard PDB, and you can open everything in whatever package you have, commercial or free packages, right? So a session looks like this. So this is UCSF Chimera. Here in purple you have the protein in cartoon, and in blue here the first docking solution, right, of our hit. You have all the information about docking in this window here. So you can simply going to one line to the other, browse all the results. So for instance, in cluster 0, there is only little variation of the different solution. But if you go to cluster 1, you see that the solution are really elsewhere, right? I don't want to go too much in details into docking interpretation. If we go back to cluster 0, and maybe we display here in orange the co-crystallized ligand of our Plasmodium falciparum target here. This is the co-crystallized ligand, and maybe we remove the protein. You can see that they are located at the same place, but the binding mode is not exactly the same, right? A lot of different possibilities for that, but surely water. So in a very standard docking methodology, first you remove water and look what happened. You can see, for instance, this water here is really clashing with our docking solution. maybe if we rerun the docking, including this water that seems to make an interaction, a bridge with here the nitrogen here, most probably the docking will be more precise and the blue molecule will be overlapped with the orange molecule. But it's maybe too refined analysis here. The question was, does the docking make sense to say, yeah, it's a probable target? Here, if we simply display the surface of our peptidase, you can see that our molecule is able to be accommodated in this at the same location, in the same pocket that the co-crystallized ligand. And surely here you have gained quite a lot of confidence about this protein being a possible target for your hit you get from your screen, right? All right. If we have time, I think we have a little bit of time. The next question is how to generate design ideas, how to modify this compound for optimization or simply to make hit expansion, right? Maybe one of the deficiency of this compound, even though it's a pretty lead-like and drug-like molecules, is that it's a sphiterionic molecules, right? At pH, physiological pH, you have here an acidic, carboxylate here, and here the amine is protonated. So okay, it's not a no-go, but maybe you want to get rid of this of one of these charge for better PK or whatever reason. So one way to get idea of how to modify your hit is to make fragment-based design and bioisosteric design, right? Vincent have told you about Swiss Bioisostere. So this is the database you can simply browse with our interface here, and the database was constructed by us with all the possible fragments that were modified and tested on the same target in the same conditions, right? So let's say in the dedicated exercise here that we want maybe to get rid of this acidic moiety here and keep only the basic function, right? So Vincent already introduced to you the fact that you have to draw the molecule here. This is the acidic function, and this is the amide, right? So this is where we are, and you have to explicitly input where is the cut in your molecule, right? So R1, I don't know why it's doing this, R1 and R2. So really, this is the fragment you want to search in the Swiss Bioisostere database, and this is where the attachment point of the rest of the molecule is. Right, I think I got it right. So I query the database and get this first panel here. Right, so here, for instance, you have all the different possible replacements here. You have, for instance, this is the most frequent replacement we can find. This is the second most, the third most, and so on and so forth. The frequency is here. And you have, in the different examples we have found, which are the number of examples increasing the bioactivity, having more or less the same bioactivity, and worse bioactivity, right? Different in log P can be interesting to have physicochemical beholding of this change. So more polar, less lipophilic, also the size and other things, right? Here, as you can see, you don't have many replacement for this specific one. It's because I was not entering the right fragment, right? So I will redo it. Sorry for that. So this is the acid, this is the amide, right? So here I have to give the nitrogen and narrow down to an unsubstituted amide. This is where is the first cut. This is where is the second cut. Right, this is better. I think we are there. Right, this is what I expected. So this is the first fragment that have to replace. So a lot of differences. You can increase the size of the linker here. And on rank 3 and 4, you have interesting replacement, because here you have the typical trick of make an ester, so methyl ester. And here you have also medicinal chemistry trick of replacing the acidic function by an amide, right? If you click on a specific fragment, you have all the examples. So you have a lot of data giving you some information in this bar here. So the number of time it was found replaced, the difference in log P, and so on and so forth. You have also the analysis of the connection points, if it's connected to aromatic, to aliphatic linker, and so on and so forth. Here is the table of molecules where this specific replacement was found. So going from, in that context, going from carboxylate to methyl ester here. And you have this kind of information. So we can, there is 101 examples, so we can expand the table to get everything. And we can maybe sort already by target class to narrow down our search only to proteases. So, you know, you have a general information, but you can narrow down to the biological context, so the target class, and also to the chemical context. So if you click on every individual line here, you can see here on this specific protease that is catepsin, this molecule was tested. This molecule was tested in the same paper, in the same assay, and this one was more active, right? And the difference is going from this acid to this ester here, right? If we click on other examples, this is the same molecules, but on another proteases, and this is maybe more interesting. So you have something that is pretty drug-like. You can see here, you have an amide, you have kind of hydrophobic linker and the acid, and in that specific proteases, going from that acid to that methyl ester increase the PIC50. So you have an increase of potency here. Of course, if you want to get more detail on that, you can click on this link, which is the PubMed ID. So you have a direct access to the article or to the abstract, actually. Of course, you can go back to the first panel and browse other fragmental possibilities. So this is transformation in the amide. Again, all the details about this replacement inside the Swiss Bioisostere database. If we show the entire table and we narrow down to protease, you see that in that context, much less number of examples. You have a few. Actually, it's the same molecule as before, but on another. So the same the same class of molecules, but here instead of getting from the acid to the methylated compound, you have from the acid to the amide. So here already you can do a little SAR. You have other examples. This is purely, this was purely peptidic, but this is more drug-like. Interestingly, for the angiotensin converting enzymes, ECA, you have a drug-like molecule of this kind, and you can stay at the same level of activity going from here to here. Right. Of course, it's far from being the end of the story, in the sense that in the docking, maybe when you redock with the specific water, you have to check if this what contact is making this acidic moiety. If it's really a salt bridge, maybe it's not very a good idea to make a non-charged amide. But if it's only making some H bonds, it can be a good trick to get rid of this acidic moiety, to have something that is more drug-like with a better PK properties. So I think with that, I am done. I try to exemplify the few tools. Maybe the next step for us is to enhance Swiss target prediction with microbial targets, maybe for Plasmodium or other interesting target for infectious disease. This is something we have to evaluate if how it is feasible. So for this specific therapeutic area, you could be possibly in the future use Swiss target prediction directly with the right species. So I thank you, and we are happy to answer any questions should you have some.
Thanks for a great presentation, both of you. Yes, that was one question I was going to ask, because of course Kemble does already contain lots of information about testing and falciparum, TB testing, and all sorts of different organisms. So would one way be simply to just allow your bioactive compound library to include data from other organisms, rather than just restricting them to the mammalian species?
Yeah, technically it's very feasible to mine Kemble with, for instance, Plasmodium species instead of human species. Then a few assessments on the validity of our scoring function if we change the species, and it's related, of course, to the amount of data we have in Kemble and the quality of data we have in Kemble and the chemical diversity. Mm-hmm. Honestly, we haven't assessed this possibility yet, but we have the feeling it's fully feasible.
Mm. But thanks anyway for showing how you can use these tools to approach targets that actually aren't automatically in the databases. That was really interesting. I have some questions for you in the question box. The first one is from Mark, who says, can you see the alignments between ligand and database compound in shape-based alignment, Swiss Similarity? Can you see the alignments?
See the 3D alignment? Not directly.
Right.
No. Through the website, it's not possible. So of course, we of course have access. If we have a specific collaboration or specific need, we can retrieve ourselves the root of the alignment. But through the web-based, it's not possible. You have a restricted, if you want, option in terms of parameterization of the runs, and of course the output. It has to stay in a web-based friendly fashion. So giving all the possibilities not possible through a web browser.
Okay, I have a question from Andre Horoshek, who says, Are the molecules being submitted to any of the tools being treated as confidential? I know you answered this one last time.
So sorry, I don't get it. If the molecule have, there is some confidentiality issue with our websites.
Yes. So when people upload structures, are they treated as confidential, I suppose?
So in principle, yes, in the sense that we are not looking of the molecule that were inputted on our website. We have no incentive to look at these molecules, but technically they stay on servers for at least 1 week, and we keep trace for statistical purposes, so number of jobs and so on and so forth. There are some confidentiality issues because, of course, we are trying to be as much as possible secure on our servers, but it's not said that we will not be attacked one day or the other, so we cannot ensure confidentiality. So if you want to have pure confidentiality, super robust thing, you need a special contract with us. So companies, we are working with companies, we have contract with them to implement these tools to their infrastructure, so in-house, behind their firewalls, screening their own libraries. But of course, it's a contract. The submitting something on the web through we are not interested in what is inputted, but we cannot assure that it's totally safe.
And also the other trick is always to just put in a close relative of the compound you're interested in.
Sorry, the sound is pretty bad.
Sorry.
If
you are particularly worried about the confidentiality of your compound, you can always put in a close relative, something that's not quite the right compound. If you're interested in bioisostere of particular parts and you're not interested in the whole, you don't have to disclose the whole of the rest of the molecule. You could just invent groups.
Exactly. For some specific purposes, you can put only one part of or changing the part of your molecules you are not interested in, or put something very similar. You think it won't change so much. There are a lot of different tricks to apply. If it's really for a massive utilization of these tools with IP sensitive molecules, it's better to get in contact with us, and then it's another legal and context, if you want.
I have a question here from Chris Swain, who says Swiss Bioisostere was generated using Kemble 17. Any plans to recreate using Kemble 23?
Yep. So there is currently a plan, actually, for different reasons. We are in contact with the company, actually, and so we need to update the different tools. So Swiss Bioisostere and Swiss Target Prediction, in a couple of weeks from now, will be updated to the latest version of Kemble.
Thank you. Good question. Okay, I think that's probably all the questions I have. If anybody wants to put a quick one in, now's your chance. Oh, so I think it's for me to say a big thank you from everybody to Antoine and Vincent for all your hard work and in giving not only this presentation, but this is the second presentation, and we are hugely appreciative of your efforts. Thank you very much, guys.
Thank you very much. It was a pleasure.
Okay.
See you soon.
See you soon. Bye, everyone.
Bye.
I'll close the webinar now. Okay.